All right, thanks, Chuck. I don't think I'm going to live up to the flattery that Chuck is bestowing upon me, but hopefully be able to give you sort of a basic introduction. So this talk is I call it Overview and Examples of Metabolomics Methods. The goal here is to sort of start at the beginning and define basic terms. For those of you who are very familiar with metabolomics already, or at least even moderately familiar with metabolomics or omics sciences, you may find this talk a little bit basic level. It may not be of great use in that it may not expand what you already know. But at a 4-day workshop, we like to make sure everybody's on the same page, that we have some of our terms well defined, just so that you know what we're talking about when we move forward. So this is a workshop, so if anybody has questions, feel free to stop me in the middle. At least I'm fine with doing it that way. Makes things more interactive. So just light up your microphone. Of course, if I don't notice you right away, I'm sorry. Sometimes I get a little bit carried away moving forward. So anyway, metabolomics methods. Let's see what we've got here. As Chuck had already said, let's see if this will work. So the objectives of this session, by the end of the session, hopefully you'll be able to know what a metabolite is. Probably you wouldn't be here if you didn't have some interest already, and how metabolomics is useful. Define the basic metabolomics techniques that we'll refer to again and again, targeted, untargeted, and flux omics. And then hopefully you'll get a sense of the purpose, why you would go for one over another, and what the limitations are, what you can do and you can't do when you take a particular tack on your metabolomics data. All right, so moving forward. So, okay, so this is the workflow slide. You may see in some of our talks to beginning, from beginning to end of this workshop, you know, we like to think of it not as a straight line, but as a repeating process. One experiment informs another in science in general, and definitely metabolomics, that's no exception. But just to look at it first, so you have an idea, you consult with the core, you design your study, collect your samples, and this is certainly a very important process. There's going to be talks right after this one about what that entails, and a good way to do that. Prepare your samples, so that's getting things ready. And then we go through the metabolomics workflow, what we do here at the core, what if you were running a metabolomics lab you would have to do, and you get some data out of that. Usually you'll do some sort of statistics, you'll interpret it, and then that will inform, you'll either have your answer, you'll publish a paper and move on to another project, or that data will give you another idea and you'll move forward. So we're here, but really this workflow encompasses a lot of all of these steps. All right, so and now one more introduction to the presentation. First we'll define the terms. We'll look at different metabolomic strategies, specifically targeted, untargeted, and flux. And then I'll tell you in the course of the talk why you know, it's nice to have these nice, clean, hard, razor-sharp sort of terms, but they don't really apply as much as they used to, as Chuck was sort of alluding to. And a lot of the times there's some blurring. Still some cases why the terms are useful to know, though. Okay, so super basic. And I apologize for all of you. What is a metabolite exactly? A metabolite, and so this is our working definition for this workshop, or at least as far as my talks. I call it any small molecule, and that means it's got a molecular weight usually under 2,000, which can be found within or produced by a biological system. So that means a cell, a human, bacteria, whatever it is that you're interested in studying. So sometimes the most useful way to define what a metabolite is by defining something that is that meets the rest of this definition but is not a metabolite. So DNA, RNA, protein, these macromolecules are not what we consider metabolites, but small molecules are. So what could these small molecules be? They might be a fuel substrate like glucose or a fatty acid. They could be a structural component of a cell like a phospholipid, part of a cell membrane. They could be a signaling molecule, a steroid that circulates in the blood. There are many other classes. Metabolites also can include exogenous compounds. So it can include drugs. It can include environmental things that you're exposed to. These things can all be defined, you know, depending on your working definition for your study as a metabolite. And macromolecules also interact with metabolites. So these things tend to be made up of smaller subunits, and many of these small subunits are metabolites. So DNA made up of deoxynucleosides, RNA nucleosides, proteins made up of peptides and amino acids are certainly involved as fuel substrates and other components of a cell. So There's a lot of interaction in complex biological systems, and metabolites are in the middle of all of it. So what is metabolomics? Metabolomics then is the attempt to identify and, importantly, quantitate in one way or another all of these species, all in quotation marks. Can we really measure every single thing that is in a biological sample? No, we likely can't. But the goal is to get as close as we can, or as close as you need to be for your study. In biological samples, which are widely defined, can be almost anything that you can imagine. We've probably had someone come to the core, and if you've got something that we haven't yet looked at, we would like to help you with it. And study their roles in these complex biological systems. So not just to be able to measure these things and give you a spreadsheet with numbers on it, maybe that will be great, but we also want to put you in a position, or you want to put yourself in a position to be able to interpret something from it. Okay. So metabolomics workflow, simplified. So this is, I showed you the circular sort of slide. Now let's take it in steps. And I think importantly here, since this is a metabolomics core, a lot of the times, depending on how you're going to do your study, you may not do every step of the experiment that we're going to tell you about. And so it's good to define who does what in a typical core user sort of study. So I broke things down more basically into four boxes here: a biological experiment, sample preparation, getting your biological samples that you collected ready for metabolomics, instrumental analysis, the proverbial black box that we'll talk a little bit more later this afternoon, always a fun one for me, and then data analysis and interpretation, so figuring out what it means. And so usually, traditionally speaking, you will wind up doing most of your biological experiment. You probably have some hypotheses that you may be interested in testing, or you may want to develop a hypothesis about something that you're trying to study. However, we would like to be involved to the extent that it's appropriate in helping you design a biological experiment that's suitable to metabolomics analysis. Not necessarily everything you can envision that might work in other contexts of other studies will be a good metabolomics study, and so there should be some interaction between you and us, the core, or you or whoever does your metabolomics studies to make that work out well. Sample prep generally is done in the metabolomics core. As far as samples get submitted to us, we process them, get them ready. However, sometimes there's steps that you need to take in planning your study to make your samples ready for metabolomics, and so we'll talk more about that in later sessions. Instrumental analysis, as far as the classical workflow, is usually done by us here in the core. Now, if you're going to go and set up a metabolomics core, we've had people at this workshop or a metabolomics lab that's what they're interested in. Then you'll want to pay attention to the instrumental analysis talks with a keener ear. But for a lot of you who are going to be submitting samples to a lab and having them analyze, you need to know about what's going on so that you can make sure that you are able to understand why we're doing what we're doing, so that you know how to interpret your data when you get it. But generally, the instrumental analysis, running the samples, is handled by us. And then data analysis and interpretation, oftentimes that's where the things transition back to you. We can either hand you a table with a spreadsheet with numbers on it that you'll be ready to interpret, or it can be more interactive. It can be, especially in the context of untargeted metabolomics data, if you're new to the field, there may be some need for some interaction. What does this mean? What do we think about where this is going to be useful? And how does this inform my next steps in the study? Okay, so Chuck already showed you the slide. You probably already read it when it was up on his screen, but I think it's worth taking some time just to define these terms: targeted metabolomics and untargeted metabolomics. What makes a targeted and an untargeted study distinct? What make them the same? And eventually, how are you going to pick between them when you have to start an experiment? So targeted metabolomics is what you do when you already know what you're looking for. You come to us and say, I'm interested in studying acylcarnitines in the context of diabetes, and I want to measure acylcarnitines in this blood sample or amino acids in this cell culture. So if you know what you're looking for, a particular class of metabolites, then targeted is a good place to go. Targeted metabolomics usually measures tens or low hundreds of compounds. There's some finite limit about how many species you can look at, and oftentimes it winds up being defined by the class. We'll look at a particular class, be it amino acids, be it acylcarnitines, be it more broadly speaking, lipids, be it very specific, a single drug and maybe a metabolite thereof. It's a good way to go for those sort of studies where you already know what you're looking at. One of the characteristics of targeted metabolomics is it produces data with confident identities. So when you submit your sample, you get data back. You have a molecular species attributed to every quantitative value. So you know exactly what compound you're looking at. And the reason is because we've tested the method. We've validated the method using authentic standards. We've purchased or had synthesized a pure chemical compound. We've run it on our method. We know exactly how it behaves. We can identify that compound in your sample, and so there's no ambiguity there. And you can get either absolute or relative quantitation. So we can give you micromolar concentration, nanomolar concentration in a sample, concentration relative to amount of tissue, or we can, if it's efficient, we can use relative quantitation, fold change in one group versus another. An example would be free fatty acid profile of blood serum. Okay, so that's targeted metabolomics. Untargeted is maybe what you might think of the true discovery omics sort of science. And so if you don't know what you're looking for, if you have a condition that you're trying to study, a treatment and control. Yes, question.
Sorry, I had a question.
Sure.
Go back. When you're talking about authentic standards, are you meaning standards that can be created in the lab, or are you talking actual proper standards?
So we're talking about, so that term authentic standard is a little bit, of a, I don't even always like it, but it's frequently used to differentiate. It's something that you might purchase from Sigma Aldrich. It could be synthesized in a lab as a pure compound, but it's not something where there's any degree of ambiguity, like say you were to culture yeast cells and extract something from it, and you thought you probably had something based on the information, but there had been no independent validation. An authentic standard is something that, like, you would buy from a lab with a label on it. You know what it is, and it's been validated by some external source. So we know for sure what it is. It's not a standard that you developed and you think you know what it is. Does that basically answer your question? I think I used more words than needed. There may be some other, fancier, reason why some people use authentic standards as opposed to, there's other classes of standards. There's internal standards. There's isotopically labeled standards. We'll talk more about those other things in various other sessions. So, okay, so back to untargeted. Any other questions before continuing on? Okay, cool. So when you're trying to figure out what's going to go on in an experiment, you have a treatment and a control. You have some sort of a manipulation that you're doing, and you want to understand what might be going on in metabolism, but you don't know exactly what pathway might be affected. Untargeted would be a good way to go. Untargeted tends to measure more compounds than targeted metabolomics. Depending on your sample type, it can measure up to thousands of compounds. The identities, however, are not always as certain as what you get out of targeted, classically speaking. So some compounds in an untargeted data set produced by the Metabolomics core will have confident identities. You'll have a list of species with quantitative values that you can look at, and you'll know what these things are. But some compounds out of an untargeted data set may not be identified at the outset. You may have a list of unknowns. Sometimes we call them known unknowns, things we see frequently in a particular data set. We can define that they're there. We know they're a metabolite or a feature of a metabolite in the data, but we don't have a name assigned to it, at least not yet. And so whether they're important or not can be determined by whether they are relevant to the groups that you are trying to study, and that can inform whether you actually go after figuring out what these things are or not. Generally, untargeted metabolomics is limited to relative quantitation, things like fold change or some other quantitative metric, which will give you an idea of how things differ between groups, but it won't necessarily be absolute quantitation, a micromolar concentration. And so if you were looking for a global study of metabolic response to a change in diet, that might be a good example of an untargeted study. Okay, so spent a long time on that slide. Important to look at in general. You'll get a much better sense for how all of this works in this talk and then in future lectures by Maureen and others later on in the workshop. So let's look briefly at a targeted metabolomics basic workflow. So first steps are sample preparation. We take your biological sample, be it a cell, be it a tissue, be it a biofluid, and we get it ready for metabolomics. So we take Whatever your matrix is, we extract the metabolites, ideally remove the things that we're not interested in, like proteins, macromolecules that might otherwise interfere with the analysis, and generate some sort of an extract that's ready for usually some sort of a solution that's ready for instrumental analysis. And we'll talk more about that later this afternoon. Instrumental analysis in our lab is usually done by liquid chromatography mass spec or gas chromatography mass spec. It will generate data, and we'll look at that in just a couple slides. The data usually comes in the form of chromatographic peaks. We analyze the data to get the quantitative information out. If we're doing absolute quantitation in the targeted workflow, we will do some sort of a calibration to tell you how much signal corresponds to what concentration of metabolite, and then you get your data. So very straightforward, as if you were doing a biochemical assay, just with more compounds on the output. Okay, so sample preparation. The goal of sample preparation is to make a biological sample ready for analysis. And so the typical steps that we will do in the lab, or that you might do if you were doing preparation yourself, would be to add some sort of an extraction solvent. Extraction solvents in metabolomics are a little bit different from most other macromolecule sort of studies. Often it will contain or be primarily composed of an organic solvent. Methanol, acetonitrile, chloroform are typical solvents that might be used. The reason for that is because metabolites tend, if you maintain sufficient polarity, metabolites tend to be reasonably decently soluble in a lot of these solvents, as well as their native aqueous solution that they're going to be in a biological system. Whereas macromolecules, proteins, DNA, these things are less soluble or not soluble. So you'll remove your macromolecules while leaving your metabolites in solution, and that's what we want. So it can be more complicated than that, but that's often the basic step. Extraction solvent can be 1 or 2 phases. You may just have an organic phase, you may have an aqueous and a separated organic phase, and you may pick one of those two for your downstream analysis. Your extraction solvent may contain internal standards, something you add to the solution to be able to follow the extraction through the steps to say how much of my metabolite did I recover, how much is there when I go to do quantitation. And so then the goal is that you remove these macromolecules and leave your metabolites in solution. Okay, sure. So generally we consider lipid. Sure, absolutely. So the question is, so lipids are somewhere between small and big. It's not glucose. It's not an amino acid with 100 Daltons. It's not a protein with a molecular weight of 30,000, somewhere in the middle. Generally, I am using the term of metabolomics to contain lipids. Lipids are usually under 2,000 Daltons. There may be some extremely large species, cardiolipins, or some sort of polymers that get bigger than that, that would be over that classical definition, that might not behave well on most of our methods. But in general, we consider lipids to be like a metabolite. Now, when you hear about metabolomics, you'll hear about a distinction that metabolomics sometimes is used to refer to studying polar compounds, whereas lipidomics is the nonpolars. For the purposes of this talk, I'm using metabolomics, metabolites as an overarching technique to define both polar metabolites and lipids. Another question over here.
Yeah. So what do you do if you have analytes that bind to the protein?
Sure. So it turns out that this organic solvent that I was talking about helps you with that a lot. So oftentimes, say you have a free fatty acid, very typically in blood serum, free fatty acids are almost entirely bound to serum albumin, right? So the organic solvent actually disrupts that interaction, right? A lipid like a free fatty acid is not going to be too favorably solubilized by water or a salt solution. But methanol is another story. So it's actually much more soluble there. So a lot of the times that organic solvent will disrupt the interaction between your hydrophobic metabolite and bring it into solution, and while precipitating your protein. There may be occasions where you have a really strong interaction that needs to be broken down, and we may have to do some sort of chemistry in the process. We may have to add a reagent that particularly disrupts that. But by and large, it's not very common. Most of the times, the extraction is enough to get you a really reasonably good coverage.
Right. So in the case where it actually does not disrupt it, like the typical organic solvents, like methanol, acetonitrile, things like that, what would your next step be? Or should
So,
I talk to you about it after?
so yes, maybe we should talk offline about details. I'm not familiar with a whole lot of circumstances where that would be the case. You might consider some sort of a lysis sort of process. You might consider an enzyme that would break down whatever your protein is while leaving your metabolite intact. You have to be real careful about those sort of conditions because metabolites, as you'll hear in upcoming talks, are very sensitive to active enzymes remaining in solution, right? So you might be able to take DNA and extract it and leave it room temperature for a while and everything will be fine. Metabolites, you leave them in solution for 5 minutes in aqueous solution, and the enzymes that are free in solution have gone crazy and disrupted your metabolome. So we need to be very careful about those sort of steps in there. So probably talking offline about your specific circumstances would be a good idea. Okay, moving forwards, sample prep. So after you've extracted your metabolites, great, we've gotten rid of our macromolecules, what do we do next? Well, one option is just to analyze it as is. Sometimes our organic solvent containing solution is ready to be put on an instrument, and that's great. It's nice and clean. Oftentimes that doesn't work out. So sometimes we want to we make a dilute solution of our metabolites with this organic solvent. We want to get a more concentrated solution in a solvent that's more amenable to analysis. So we will dry down, remove these organic solvents in nitrogen or under vacuum, and then we will reconstitute it in a different solvent, perhaps in a smaller volume, and then we will take that and analyze it. Sometimes that's not even enough. Sometimes we need to further purify. Maybe we're looking at a very low abundance metabolite, and we need to take what we've gotten from our extract, and we need to use solid phase extraction to really pull down a particular class of metabolites that are present but at a low abundance, or maybe there's something else that's interfering. So we could do further purification. Sometimes we need to take these things and we need to chemically derivatize them. We need to use some sort of a reaction to get them ready for instrumental analysis. That's very frequent in the case of GCMS, where everything needs to be in the gas phase. We might have to do some sort of a chemical trick to make it more volatile so that we can put it on our instrument. So all of these things, the core lab will help you do these sort of things. If you're designing your own study and doing your own metabolomics, you need to be aware of the many possible ways. Okay, so for those of you who are submitting samples to the core, how does this affect me? The core is going to deal with it for the most part. Do I even need to worry about it? Well, yes, it's usually handled by the core, but good sample prep requires good sample collection, right? So if you don't collect your samples in the right way, you're not going to be able to get good data out of it, no matter how well we try to extract your metabolites. Metabolite turnover is a big problem in aqueous solution if you're not carefully managing it or minimizing the time of exposure to it. So sometimes you may need to do some of the sample preps yourself, so you need to be aware of those. And also method development, which Steffi will talk to you, I think, a little bit later on. That's a new add-on for this workshop, is sometimes needed to determine the optimal sample prep strategy. So sometimes that's an iterative process. So the take-home message is please consult the core before beginning this experiment as early as you can. Okay, instrumental analysis, the second step, we're only on step 2, is to use to detect and analyze these metabolites in your sample. Is it a black box? No, it's not. We'll try to open up that black box later this afternoon. Okay, not a black box. Okay, analyzing the raw data. So we'll talk a little bit more about this later on in other sessions, but the goal is to take the raw output, a chromatogram, some other form of data, and pick out the signal that comes from individual metabolites. And so the output of a LCMS experiment is a chromatogram, a plot of intensity versus time. And we can differentiate the signal that's coming from different metabolites based on mass. This is we'll talk a little bit more about how exactly that works this afternoon. But a very useful feature of metabolites and all biological molecules, and every molecule for that matter, is everything has a mass. And a lot of the times, the masses tend to be fairly unique. In the case of carnitines, we can see the individual masses for all these different types of carnitines. That's actually a misprint there. That's incorrect. Sorry, that should not be 204. Although we do sometimes see things which have the same mass. C2 carnitine and C4 carnitine actually do have different masses. And then from this signal, we look at the area under the curve to get the quantitative estimate, and then we have a table of metabolite abundances. This is not yet concentration values. If we want to go further, we can do some sort of calibration, not unlike a calibration you would do with any other assay, except it's using mass spec data, and we have a separate calibration for each metabolite based on the peak area, perhaps an internal standard peak area, rather than just using the ratio of the I'm sorry, the calibration between the measured peak area and the concentration. We might use the ratio of your metabolite to an internal standard peak area. And then we will generate quantitative data from there. And then it's data analysis. And so this is where it should be more familiar in targeted metabolomics. You may be able to interpret a change in metabolite concentration without any further ado. That may be enough. You can do basic statistics, a t-test or an ANOVA, perhaps some multi-comparison correction, and it's as easy as that. A lot of the times it's more complicated. Okay, so untargeted metabolomics, how does it differ? Sample prep, very similar. Maybe need a few more standards to make sure we can monitor things properly, but basically the same steps apply. Instrumental analysis, also very similar. The unique part of untargeted analysis really comes in how we manage the data. The choice of instrument, also important for untargeted analysis versus targeted. We'll talk more about that. So the first step in untargeted analysis, we'll talk in a little bit, is feature detection. Rather than saying, okay, I have a standard, I know where it comes out in my method, I'm going to quantitate it, we want to say, what's everything in my sample? Let's find everything that could possibly be a metabolite, and then let's look at it and see if it's of interest. Okay, and so then the next step is feature alignment. We want to find not just all the features in all the samples and say we're done. We want to say which features are in common between samples, and align those so that we can put data from a single compound, ideally a metabolite, in the same column and compare them from one sample to another, from one group to another. Sometimes there's multiple steps that take place here. Recursive processing. We found some features in some samples, but not all samples. Are we sure that the computer, which really did this feature detection for us, did a good job finding all of our features, or can it do better if we say, go back and look for things that you missed? Then we might do data analysis. So without necessarily having identified anything yet, we can do statistical and multivariate analysis or univariate analysis to say what was different between two groups. And then we'll use that and say, okay, give me a list of those features that are differentiating between groups. And then, and only then, let's go and figure out what those features are. Some of them you'll already know because they were already given to you in a table with identities. Some of them may be unidentified, and there may be more work required to figure out what they are. But that's the point where you really want to go and invest the effort in identifying the unknowns. And then you go and do further analysis, which will be a major subject. Okay, so already said sample prep and instrumental analysis, very similar. Feature detection, as I mentioned before, is where we attempt to find all the signals. This is performed first on each chromatogram, each individual sample, one by one. And so we just try to find everything that looks like a peak, looks like signal that could be coming from metabolite in all of our data. And then we go through and align them, as I already said. And the output of this, so we find features and we can classify them by group. It results in a table with mass and retention time data. We'll talk more about that when we get into the details of mass spectrometry and chromatography. But a table of features with their relative abundance in each sample. And then we do data analysis. And as I mentioned, it can be multivariate or univariate analysis. Principal component analysis or other methods are commonly used, which detect features that differentiate the groups. And now we have a list of features which are potentially important to your biological study. So you'll want to be looking at your treatment versus control, at cancer versus normal, at whatever other comparison that you're interested in. Sometimes it's multiple groups that you're looking at. And then, like I said, you're going to go and try to identify these features. Usually that can be a complicated process. It may be done by searching against an online library of metabolites, or it could be it could require additional steps. It could require follow-up analytical steps where we will take we will go and purchase a standard that we didn't have in our library and try to figure out if it matches with the data that's coming from your sample. It might even require synthesizing a new standard if it's something that's really more uncommon. But basically, the goal out of this process, which is sometimes successful and sometimes partially successful, sometimes unsuccessful, is to get a confirmed ID, or if not that, a putative ID, depends on how far you want to go and how much time you want to spend, that is a candidate or candidates for the identity of that metabolite. So this is to give you a sense, not that, oh my gosh, you're telling me that we're not going to know what comes out of an metabolomics experiment and what good is that to me. A lot of the times, you'll be able to use other data in your study to help you in this process of identifying your metabolites. Or you'll be able to narrow something down to a class. You might say, okay, I have a phospholipid. It looks like it has this chain length, and it's of this class. That's good enough. I may not need to go and identify exactly where my double bonds are in that species, and you'll get fairly far. So this is to make you aware that there are these complexities of untargeted data analysis, but we're here to help you manage those complexities. And then the final step is to interpret the data. And this is true in untargeted metabolomics as well as targeted. You may go and visualize the data as a network, as a heat map to look at connections, correlations between metabolites, or you may use other tools to help you extract biological meaning. Okay, so now back to basics. How do you pick, right? How do you decide which you want to do when you're starting a study? Do I want to do an untargeted study? Do I want to start in at a targeted level? How do I decide? So for a new project, when you're beginning, a workflow beginning with an untargeted analysis and leading to targeted analysis is often best. When you just have a hypothesis, or when you're not sure and you want to compare some samples that you've got, and you don't already know exactly where you want to be looking, oftentimes an untargeted study is the place to go. And you'll get some really good, very specific metabolite quantitation out of the untargeted study, which may be enough, or it may lead you to a targeted study. Some applications, however, will favor one of the methods over another. So if you're trying to do biomarker discovery, that usually would mean that you're going to want to follow an untargeted workflow. If you're trying to generate hypotheses, usually that's an untargeted workflow. If you want the broadest coverage possible, if you don't want to leave anything out of your study, untargeted is a good way to go. Now, if you were doing biomarker validation, if you had a candidate that you wanted to study, and you wanted to make sure that this was really significant, then you might choose to do a targeted workflow. If you wanted to quantitate metabolites very exactingly, targeted would be a good way to go. If you need a high certainty of your identities of all metabolites that you're studying, targeted would probably be a way to go. And if you need absolute quantitation, you're pretty much going to be locked into some sort of a targeted workflow. Okay, so I've spent this whole talk defining the differences between targeted and untargeted, and now I'll tell you that I used to give this talk and that was it, right? The line was fairly hard. The instrument determined which workflow you were going to follow. But in reality, as the field has progressed, the line is blurring between untargeted and targeted metabolomics. Here's a paper written by one of our colleagues on the West Coast, Oliver Fien, called Toward Merging an Untargeted and Targeted Metabolomics Methods. And so it turns out that a lot of data that you get from what would classically be considered an untargeted workflow is very valid for targeted metabolite analysis. And so a lot of this has been brought about by improvements in sensitivity of instrumentation and better ways of analyzing the data. So a lot of the times an untargeted study can be mined in a targeted way and can give you really good quantitative data. Some workflows remain a pretty classically targeted study. If you decide that you need the best sensitivity possible and you're going to generate, and we need to use the instrument that has absolutely the best possible sensitivity, that'll probably lead you to something like a triple quad type analysis. I'll tell you what that is later if you're not familiar. Basically, you'll need that workflow, but the nature of doing the experiment will lock you into a targeted workflow. And really what you need to do is talk with the core to understand the study. Okay, one more term, which we'll talk more about tomorrow. I know I'm already over my time. Metabolomics versus fluxomics. The conventional metabolomics measures only the concentration or relative abundance of metabolites. Flux, however, the rate of turnover of metabolites are very dynamic, is also a really important aspect of cellular metabolism. And you can't measure flux. You might think that changes in concentration would tell you something about flux, and sometimes you would be right. But flux itself, the rate of turnover, cannot be measured by just observing how much of a particular metabolite is present. And so what is flux exactly? You can kind of think about it like the flowing water through a stream. You have metabolites that are the source, maybe an upstream pool. You have the flow between pools, and then you have metabolite pools. So the volume of this pool would reflect the concentration of a metabolite. And then you have flow out of a pool. You have a source and a sink. In general, the amount flowing into a pool should be equal to the amount flowing out of a pool. And if it's not, one of the pools is growing and shrinking, so you're not at steady state. and that can certainly happen. The size of pool is independent of the amount of flow into and out of the pool. So that goes back to the fact that just measuring the size of this pool doesn't tell you how rapidly the water is flowing. Measuring how much of a metabolite you've got doesn't tell you how rapidly things are turning over. So we'll talk more about ways to measure flux in a talk tomorrow. The question is, do you need to be concerned about flux right from the get-go? Well, for a new project, I would say starting out trying to measure flux and concentration is often too much complexity. You may be better off starting with a targeted or untargeted conventional metabolomics workflow, the outcome of which will often inform whether you need to measure flux, whether you need to measure the turnover or not. But exceptions do exist. Sometimes we can tell right off the bat that you're going to need to have some study of flux, and we may start you out with that type of experiment. Okay, so in summary, metabolomics is a study of the full complement, in quotation marks, of small molecules. Targeted metabolomics quantitates selected metabolites or classes thereof. Untargeted offers more comprehensive coverage, but with some trade-offs. Flux Omics, stable isotopes, we'll talk about those tomorrow, to study turnover of metabolite pools in biological samples. And the best way to select, if you're not familiar, if you don't already know, is to talk to somebody in the core or a metabolomics expert and get an idea of how your study would best be done. And that's it. I know I'm already over time. I'm happy to answer questions now or later.
Sure. So we have time for 1 or 2 questions now, and I'm going to suggest that after this we take a 10 minutes break, and you can catch up with Charles and stretch and so on and so forth, and then we'll get back here. 1 or 2 questions? Is there?
Yeah.
In the untitled workflow, do you use technical replicates instrument injections?
Good question. So sometimes yes and sometimes no. So we will always, so repeating the question. So the question was, in untargeted analysis, or targeted for that matter, do we use technical replicates of samples to assess reproducibility, et cetera? So quality control is an important part of any metabolomics experiment, and it's always essential to have some form of technical replication within a study to get an idea of how reproducible your data are. Whether it's necessary to run every single biological sample 3 times or 5 times, oftentimes it's not. Oftentimes you may take a quality control sample or a pooled sample and run that multiple times over the course of an experiment, but actually only run your biological samples once. There may be occasions when you have a very limited number of samples and you're trying to do a very low-level analysis and you actually do benefit from doing multiple replicates. Oftentimes it's more important to have biological replicates in an experiment than technical replicates. So we would never suggest that you collect a single sample, a single plasma sample in your control in an experiment and try to get data out of that. That just won't work. You know that already. So biological replicates can often give you that information about reproducibility on a biological scale and encompassing the analytical reproducibility, and our technical replicates of a pooled sample will give you the rest to say how good the technique was doing. So. Mm-hmm. Yes. You don't always have to do the technical replicate, and the core will make sure that if you provide us with a QC that you want to use in addition to your samples, that's fine. The core will always will always make an appropriate QC, sometimes multiple QC samples to analyze with your batch. We may generate a pooled sample, a combination of all of the extracts of all of your sample set. We may run an additional quality control sample, like a pooled human plasma sample that we have in our archive, so we can compare it with other assays, other methods. So it's a very important part of any study. But oftentimes more important for you to worry about your biological replicates than the analytical reproducibility. That's